Maybe we can hand it over to Ogunro.
Okay. Can you see my slide?
Yes, we can.
Okay. So hi, everyone. Thank you for signing in. Really appreciate that. So I will be talking about the IOMB. It is actually a community effort. There are a lot of people behind this. So it's, if you've heard about ILAMB, this is the ocean component of it. That it's mainly designed to evaluate marine ecosystem, and mainly I could say designed to actually evaluate the ocean model in general. Okay, so it leverages on ILAMB's code, but tailored towards benchmarking ocean model. And then you will hear the term ILAMB or IOMB tossed around a lot, and we love to really define exactly what we're talking about when we start talking about ILAMB or IOMB. In this case, the IOMB could mean the community of people, the scientists coming together to look at the benchmarking of the ocean model. The model is getting more complex, and really need to look at, evaluate the processes and the variables coming out of this model. It could actually refer to the data set, which we put together, make them CF compliant, and we have a link of all the data set we put together, and we keep updating this data set. I'm going to show the link in a couple of slides down the line. And just to, speaking of data sets, we'll be glad to get more data set from you folks. I mean, if there's, if you have any data set that we could use to evaluate marine ecosystem in the Arctic, it would be great to incorporate them into IOMB. And then IOMB could also refer to the method in which we're using. And this is part of this though is well defined in Nate's paper. I'm gonna show a link to it at the end as well. That paper came out last week, which some of the methods that serves as the fundamental of ILAMB and IOMB were well defined in that paper. So IOMB could also mean the software, and at the same time, it could mean the results. So as I said earlier, this is mainly tailored toward marine biogeochemistry, and most of the data sets we'll be using could be global, could be point data set, it could be regional. What we try to do is to score different models based on how they perform with respect to different observation data sets. So, and this is built in Python, it is open source. So if you put your attention on the two panels I have on the right-hand side, the one on the left, you see for both, you see the models listed across the top over there, and then from top to bottom, you see different variables. So the one on the left is the absolute score. I will show you the next slide or two, how we came about the absolute score, and the one on the right-hand side is the relative score, which gives us the idea of how well a particular model is doing with respect to all other models which you have lined up for that particular run. So with the green, you begin to have an idea that this model is doing well with respect to that observation data set. One thing we like to clear up front is this is not an impression that this model is good or this model is bad. It just gives us an idea of how a particular model is doing with respect to your observation data set. So the quality of the observation data set in question is very important. Talking about the overall score now, one thing we try to do is score the bias of the model and the benchmarking data set, and then score the RMSC, and then we score the spatial distribution as well. And then we put more weight on the RMSC. Again, all this is well explained in Nate's paper, which I'm gonna put a link at the back of this, at the end of this description. It's gonna go through everything. Unique features of the ocean. We are aware that the ocean is different from land. A couple of things now. The first one is for the ocean, the observation is sparse. And then we have to worry about 4 dimension in the ocean. We have to really worry about the depth, brings about the memory to run all these things. And then for the ocean, you have a lot of things moving around. So we have to worry about the physical circulation and the biogeochemical variables as well. And then one thing to also mention is that for the ocean, most of our data sets are climatologic data sets. So for example, if you look at the World Ocean Atlas, you got the Takahashi data, most of our data sets in the ocean are climatological. So what ends up happening is IOMB has to go through the model and then calculate a commensurate climatology from the model before we can begin to do that evaluation. So here's the link for the data. If you're interested, you click on that. You can see a couple of data sets over there. And then if we happen to get a data set from you, we could encode that data set to make it CF compliant. I think there's a tutorial about that as well. So basically what we require is the time, the longitude and the latitude, and then give us the bound of it, just to make it like a basic CF compliant data set and NetCDF, so that we can use it to evaluate the model. So I'm going to walk you through these 2 links very fast. The first one is the old one, but just put it in this morning. I think we may get some information out of it. So it's still the same. This is the landing page. So when you run IOMB, this is the landing page you get. Across the top, you see different models. Apologize, you see Acme over here. I know it's E3SM now. And then from top to bottom, you see different variables that you're looking at. So if I click on the result table, begin to see the variable score, which the overall score, which I talked about the other time. And then you could click on whatever variable you're looking at. In this case, I click on chlorophyll. You see, I'm using 2 data sets here. I'm using the CWIFT data set, and I'm using the GLODUP data set. One thing you notice over here is if you're using a single data set, there's a weighting method where you're using a single data set. I mean, the 100% weight is on that data set, but if you're using 2 data sets, it's kind of like weigh them equally. And yeah, in this case, I'm using 3 data sets, so you kind of like weigh everything equally, but that is something you could control based on how confident you are in that data set. You could weight 70-30. That is something that the user can actually play around with. Then you could begin to dig into each of these data sets to see what exactly is going on. But I won't do that here. I will do that for the next one. This is for the spin-up E3SM. I just want to show a couple of things. This is CSM with POP, this E3SM with POP, the old one, and then this is E3SM, I think, with MPASO, the spin-up. So I'm going to do the same thing over here. You click on the results, just limit my playing around here to just the World Ocean Atlas for nitrate. So if I click on that, then you begin to see what exactly is behind the wall here. So this is the benchmarking, the surface mean. So this is mainly for the surface. Sorry, I need to mention that this is mainly for the surface. But if you look over here, you see the profile. We kind of have a score for the profile as well. So I have a profile score over here. So if you're running model which is limited to the surface, say for example, you run out the semi-pipe model, so there will not be any score for the profile. So we have a score for the bias and then the score for the RMSC and the spatial distribution, and then we generate the overall score. So one thing I like to do, because this is mainly for the Arctic, is, so we have globe over here, but you could click on this and then look at all the drop down. So you could say, all I'm interested in is just the Arctic. Then that gives you exactly what is going on in the Arctic. Let's see if I can reduce this. Oh, I'm sorry. Okay, so that gives you an idea of what is going on in the Arctic. And then I have 3 different models here, so I can begin to click on those models, say I'm interested in this. You could see what exactly is going on in your model of interest. But one thing I would love to do is if I go to all models, this is for global, and say all I want to see is mainly for the Arctic. And instead of seeing the period mean, I just want to see the bias. So you could begin to work around this based on the science question you want to answer and based on exactly what you're interested in. This is something you could manipulate, play around with, and begin to try to adapt it based on your science question. And I think there's the data information, so you could see some of the data stuff over here. Basically, what else? You go back into the model, our model, Arctic, and then, so you could see the RMS, you could see the profile as well. Okay, you can see the profile. So this section, we're currently working on this section as well. But the main point is that you could go into this and begin to look at maybe the profile or the region of interest, in this case, the Arctic. Okay. I just decided to show these are two different regions I'm showing over here. This is phosphate for 3 different models. You could look at the stereographic projection for this model, and mainly in the Arctic. The science prospect for IOMB is mainly to explore Earth system feedbacks here with ocean carbon cycle, and then mainly to partner with E3SM to assess MPAS, Marlboro, and then CSM for more Marlboro, and then for RASM in this case. I think I showed RASM previously. So, and then continue to develop new benchmark data sets from synthesis. So this is something that is very important for the Arctic because for the, say for example, for the chlorophyll data set, seaweeds, we have limitation because of cloud. So if there's any form of data sets which you have that could help us to evaluate ocean model processes in the Arctic, it would be great to add them up to IOMB. And then we continue to develop different kind of benchmarks. Here's a list of some of them. I'm just going to end up with this take-home message that International Ocean Model Benchmarking Package provides a platform for modeling and observation community to kind of employ data integration and develop standardized observational-based metrics for model development. And the community contribution is highly welcome in the development of IOMB. So here's the tutorial over here, and here's the link to Nate's paper. So it would be great if you have any question or comment, and you could pass that along to us and we could address it. And again, so this paper mainly talked about the methodology for land. The similar paper for the ocean is on the, it should come out soon. And we're working on that currently. And I will end over here. Thank you again for tuning in.
Great, thanks, Olu.
Yeah.
It was really, really cool. Really nice soft story. Well done.
Thank you.
Are there any questions?
Yeah, I have a question here. What I have found very hard to when you know, calibrating or looking data compared to model output is very often the data have themselves a very large uncertainties. If you take sea waves, if you take fluorometric measurement of dissolve of chlorophyll, or if you take HPLC, you will have a very wide range. And what you are trying to fit is your data in between. So in your data collection and your software, where is that information available? Which data and the uncertainties in the data?
So that's a very lovely question, and that's something we get all the time, and then we keep thinking about all the time. So most of the data sets we have now actually do not have some level of uncertainty associated with them. And then we say it would be great if we could have this form of uncertainty or certainty associated with this observation data. So currently what we do is to have some form of weighting method based on is the data published? Has it been peer reviewed? I mean, is it generally acceptable in the community and things like that? So we kind of look at expert opinion in terms of how they view this data set. And that can now come into the weighting method in which you use. So as I showed, by default, IOMB is just going to weigh the data sets you put in equally. If you put in two data sets, it's just going to weigh 50-50. But if you're using, say, for example, a World Ocean Atlas and maybe a data set that has been, I mean, it's well known in the Arctic and you know that this data set is really good, you can say, oh, I don't really trust World Ocean Atlas. You can tune that down to say like 30 and then put the other data set like 70. I mean, that is could be easily done by the user. So that is how we handle that currently. Did I answer the question?
Yeah, I mean, partially, because I mean, the thing is, you're gonna probably from one project, you're gonna group them by project, right? Or are there gonna be like even like if I take the IceCape data set, for instance, that would be
right.
one data set. They have the fluorometric and they have the HPLC. How, but you are probably not gonna dissociate both of them, are you?
So are they different data sets or the same data sets?
Well, there are different data sets, but they are from the same project.
If they are different data sets, measuring the same thing, we could differentiate them, I mean, and then weigh them differently.
So in other words, to which level are you going to dissociate the data?
So what we decide to do is based on the expert opinion, if we realize that one of the data sets is has been proven to be more, maybe realistic is the good word to use in this case, dependable, then we could weigh that higher. I mean, currently there's no way to really handle that level of uncertainty for the observation. We rely on the data provider to kind of give us that information. But if that information is not included in the observation, we just weigh it. Did I?
Oh yeah, you answer my question, but
yeah, I mean, I understand perfectly. It's a big challenge. Yeah, it's something we keep thinking about, but there's no way we could handle that because I can't really assume some level of uncertainty for the data set. I mean, we kind of rely on the data provider to provide that. But unfortunately, in most situations, we don't get any form of uncertainty with the observation data set.
Yeah, it would be something that, you know, if we ever, you know, get a workshop where we bring modeler as well as the experimenters to have a session on observation uncertainties, because if the uncertainties is as large as your model uncertainty, then how, if you try to fit your model that uncertainty, where do you fit it?
Yeah, I completely agree with you. There are a lot of things that I think the ocean community needs to talk about with respect to how we use this observation data set to evaluate marine biogeochemistry processes in the model. So bringing the two communities together in a workshop would be so awesome. I think that would be great.
Olu.
Hi, Nicole.
Hi. What is? It sounds like you've progressed quite well. What is your time frame for having this package in the E3SM workflow?
I know. So Nate might be able to answer that question. As far as I know, so I've been. I spoke with Matt a couple of months ago, or maybe weeks ago, in terms of just looking at the results and then be able to evaluate it based on the spin up that they just got out. But in terms of putting it in the workflow, Nate might be able to answer the question on that. I know he was working with someone in CSM and NCAR, but for E3SM, I'm not super sure. Nate, could you comment on that?
Yeah, I haven't spoken with anybody directly about the ocean workflow part. I mean, there have been efforts to wire it into some aspect of the workflow. I'm not on that project, so I'm not familiar with exactly how it works. But I know that it has been, at least in the land part. Now, whether or not that's accessible also from oceans, or if I'm sure there's extra work that has to be done for that. So I'm not really sure. Do you know who it is we would need to talk to make sure that
maybe Forrest is aware. Forrest, are you aware of anything?
I know it's a goal to get it implemented in the E3SM workflow. You know, that model is just very recently available, and so I'm hoping that whoever is in charge of that activity now in phase 2 of the project will be able to integrate it. The code's all available. It's open source. It's available out on a Bitbucket repo. So, you know, anybody can download it and use it and mess with it and give us feedback on how you think it should be improved. So
I can tell you that right now it's Matt and I that are the workflow for the E3SM team for the Marine BGC. So I guess it's us getting access to it and learning how to use it would be a way to get it into the E3SM and get it used in the simulation. So we'll have to talk and do some collaborating, I think.
Yeah, so I would be happy to, we have some tutorials and things that we give that go and show. The ones we've given at present have been more land focused, but it's really the same code base. So it would be very simple to adapt it to oceans. There's just some extra things we would talk about. So, you know, at some meeting where it would be appropriate, I'd be more than happy to appear there and give a tutorial to help, you know, instruct the community if you guys deem that as something valuable.
That's great. Thank you.
All right. I guess we need to move on, and we'll transition to Nicole Jeffery, who is our CIS biogeochemical modeler here at Los Alamos National Laboratory. See you. Okay, cool.
And Nicole, you're on mute.
Okay, how about now?
That's better.
Good enough? Okay.
Yep.
Okay. So my talk is about CIS biogeochemical modeling for climate applications. So I'm going to be talking about both poles. And in particular, I'm going to talk about a project, the E3SM highlight. We're using the E3SM highlight model. So this is from phase 1 of the highlight project, with which Wilbert has said a little bit about. And we have lots of collaborators from Naval Postgraduate School, Pacific Northwest National Lab, University of Fairbanks, have all contributed to this work. And the funding support is from the RGCM program. Okay, the Highlight Project is interested in climate feedbacks with a focus on the polar regions. This is a schematic from phase 1 that illustrates some of the processes we were interested in exploring. And you can see that sea ice plays a role here. We're in the polar regions, and the biogeochemistry is in the ice and the marine system. The feedback we're interested in is the DMS production, which influences the cloud microphysics and brightens the cloud production. And we were interested in melt from the Antarctic and changes in the circulation and warming of the system. So the sea ice plays a role in this. We had a broad motivation, and that's looking at the climate impacts of the biogenic aerosol, dimethyl sulfide. How do polar marine DMS emissions change in future climates? How might enhanced Antarctic melt fluxes impact the polar marine biogeochemistry, and then what is the impact on the cloud radiative properties? The model that we're using to look at the sea ice biogeochemical role in this is a global model. So by that, I mean the criteria we had is that we could do biogeochemistry for the Arctic in the ice and for the Southern Ocean, and they're a little bit different. This model has evolved halodynamics, that is, the temperature and salinity profiles are both evolved. And in this way, we can get information about the microstructure inside the sea ice. We know something about the density of the plumes and the overturning inside the ice. And that way, that tells us things about biogeochemical tracers, how they might move up and down inside the ice. For the biogeochemistry, we have a very simple system. We have the macronutrients that we think are important for controlling the sea ice biogeochemistry, nitrate, silicate, ammonium. We also include dissolved iron because of its important role in the marine system. And then we have some other things, DMS, of course. The DMS precursor is produced by the microalgae, which is a cryoprotectant. Okay, so as we've put in this new vertically resolved biogeochemistry model in the sea ice, we've done some qualitative tests for its agreement. So this comes from a run that is fully 3D. It's a global simulation. You're looking here at a point in the Arctic and in the Southern Ocean, and I'm evolving that point over time, and you're seeing the ice grow. The colors are the algal carbon that's growing. And you can see a little bit of a difference between what's happening in the Arctic versus the Southern Ocean. The Arctic colors are really at the bottom of that ice, and that's what's been observed. You get these very strong bottom chlorophyll peaks in the bottom 2 to 3 centimeters. And so we're seeing that in the model. In the Southern Ocean, we see a little bit of a different feature. We get some peaks in the freeboard, which is towards the ice, the ice-snow interface. You get a lot of snow and you get flooding. And so we're getting those kinds of features as well. So we have some confidence that our model is doing what we would like, but that's not a quantitative assessment of our model. And so we devised a kind of a motivation 1.2, which is how does the sea ice chlorophyll and the primary production interact with our polar ocean biogeochemical cycling system? And I've got a couple things in red here that I'm going to talk about. I don't have much time. What are the seasonal patterns and variability in the chlorophyll, in the ice, and primary production? I'm going to just look at chlorophyll. And for the second point, if I have time, I'll look at the impact of the BGC on the ocean biogeochemistry, which is, of course, what everyone wants to know, and I save it for the end in case we don't have time. Okay, so for this kind of a simulation, we ignore the atmosphere for a bit and use it as forcing. So we're going to use core 2 forcing, which gives us a climate that is between 1948 to 2009, so present day climate. And we can do some sensitivities. The first sensitivity run we look at is where we do one-way biological coupling. That is, the ice sees the ocean nutrients and algae and phytoplankton, but the ocean doesn't notice the sea ice biogeochemistry. That's one-way coupling. We also look at changing the sea ice biogeochemical parameters and see what kind of impact that makes on the system. So here's an example of how, after doing a simulation, how do we turn around and then get an assessment if we're doing the right thing? The sea ice doesn't have, for the biology, we don't have satellite observations to tell us what's going on throughout the whole system. So we have to use field observations, which are very, very valuable, but you're looking at different points in this global system. I have 3 different points that I'm looking at. One is from the NICE field campaign. Then we have the Barrow, which is thanks to Clara Deal, and then the best field campaign as well. And then the bottom 2, I have picked some data from field campaigns ISPOL, and then there's one observations in the Prince Bay, which is, kind of a fast ice region. And you can see we do all right in some areas and maybe not so good in some areas. The blue line and the red are just different parameter values for the fluxes, a tuning parameter in the sea ice biogeochemical model. But we seem to be doing okay, but it's still a very, very difficult to get an assessment of how we're doing throughout the regions. So I'm going to move to the Southern Ocean because there's this fabulous database called ASPEC, the Antarctic Sea Ice Processes and Climate. This is from a paper by Miners et al. And this is 1,200 plus individual cores. Every dot is a different core at some particular time. The color of the dot is telling you how much chlorophyll is there. Red or black is a lot of chlorophyll and blue is not very much. So this gives us something else to compare is covered much more of the sea ice zone. So this is a simulation on your right now where I've picked the same year, tried to pick the same year and month as the observation. And we overestimate in some locations. If you look in the Ross Sea, we're getting quite a lot of chlorophyll in the sea ice that wasn't observed. But we also underestimate at other locations, especially on the western side of the peninsula. There's a lot of dark and red that were measured, and we hardly get any of those. If you look at the data now in terms of month, you can see that in the austral summer, the January and February, we're not doing a very good job. This is where you get a lot of these high chlorophyll values in the observations, but we're not seeing them at all in the model. Okay, so when we do climate work, we want to say something more general. We want to say something about the climatology of the system. And so what I've done here is I've taken the aspect data and I have a seasonal cycle. The black line is the aspect data, and the thin vertical lines show that there's quite a large standard deviation in that data. The red line is the model now using the same points as the aspect data. The blue line is using all of the sea ice data in the Southern Ocean. And so we do a fairly good job for a good part of the year, but then we have this bias in February and January where we're not getting those high values. If I now look at the total algal chlorophyll, recognizing that there's not very much sea ice in the austral summer, you can see that we're actually doing a fairly good job with the seasonal cycle of the chlorophyll in the Southern Ocean. Again, maybe a small exception is that summertime. Okay, now we look at the sea ice biogeochemical data like that. We don't often do the same thing with the thickness because we can use satellite and we use biomass and other kinds of things. But how does the thickness compare if we do the same thing? And what comes out right away to me is this bias in the Western Peninsula in ice thickness as well. If we do the same kind of climatology, we can see that we have a thickness bias in those summertime months. And that's in part explaining what's going on with the chlorophyll. Now, this is not a linear problem, so it's not enough to normalize our chlorophyll to correct our values, but it's important to get the right values for the right reasons. So biases in the physical system or differences in the physical system should produce differences in the chlorophyll. Okay, this is Arctic, so everyone's interested in the Arctic. Well, if I try to do the same thing in the Arctic, you get this very sad looking figure. There's no nice compact database that I can draw from. So I've been going to papers, it's been personal communication, I've been looking at other websites trying to get as much observations as I can. And you can see the observations are really limited to a couple regions. They're not giving me a broad swath of the Arctic. However, if I do the same thing and I look at what my climatology looks like, we're not doing too badly as opposed to the initial figures I showed. We're actually getting a curve, that's the blue line for the whole Arctic, that is consistent with the curve that those observations shown. Clearly, we don't have enough observations to really validate this region, or at least I don't have enough observations. They may be out there, and it's just up to me to find them, or other people to find them. Any help would be appreciated. Okay, I do want to get to this last part, because this is sort of the interesting thing about modeling, is what is the impact now of ice biogeochemistry on the ocean biogeochemical system. Okay, the surface, the upper two figures right now, you're looking at the ocean surface chlorophyll. Okay, this is in November and in January. It's a log plot. The high values are at 128 milligrams per meter squared. Again, this is just the ocean surface. Now I take simulations. I have one simulation where I have the ocean chlorophyll and sea ice biogeochemical chlorophyll, and then I subtract that from a system where there's no sea ice biogeochemistry. And what you see is this addition. There's this addition of chlorophyll, and my bottom line is cut off, but that's also the same 128 milligrams of chlorophyll per meter squared at the bottom. This additional value, same order of magnitude as what you're seeing in the ocean, that's a large contribution of sea ice chlorophyll to the upper ocean that you're getting. In the Arctic, you have something a little bit different. Okay, now we're looking at April and June. So this is during the sea ice chlorophyll peak and during kind of where the ocean biogeochemistry peaks. The upper surface, the upper plot is just what's in the ocean, upper ocean. If we, again, difference now a simulation with the sea ice BGC versus without, we get, yes, an addition in areas of the same order of magnitude as what's seen in the surface, but we also get these blue values. We get a decrease in what's going on in the upper ocean. And that's telling me that the sea ice in the Arctic is competing with the ocean system. It's not just adding, it's actually competing. And so we have a nutrient limited system we're getting drawdown from the ice BGC, and we're reducing what's available in the Arctic. Okay, so the conclusions are that the global model validation is challenging with ice field station observations, and I encourage the support towards a uniform database, because that's invaluable for modelers. We're getting good magnitude and a general behavior for ice BGC chlorophyll, seasonal cycle in both the Arctic and Southern Ocean, though it's difficult to validate in the Arctic. Exception possibly is the summer months. And we see that the biogeochemistry in the Southern Ocean enhances the marine chlorophyll, surface chlorophyll, whereas in the Arctic, we get some reductions and even a shifting of the surface chlorophyll. And the difference between those two regions is turning out to be iron. Okay, that's all I have.
Great, thanks, Nicole. I propose to open it up for one question, and then we'll move on to Georgina's presentation. And afterwards, I hope we'll have some more time to continue the discussions, but let's see that we can get through the presentations first. So one question for Nicole.
Yes, I have one. My experience with the ice biogeochemistry is that it's poorly constrained in terms of nutrient, and that will favor or not the bloom of chlorophyll in the ice or not. In addition, very often the grazers are not doing their job in munching what is in the ice or in the bottom of the ice. So do you have any idea of what your grazers are doing and how your nutrients are calibrated in the ice?
So I do see that the ice tends to be nutrient limited. The two different simulations we were looking at for sensitivity changed that flux from what's in the ocean up into the ice. And in the Arctic especially, when you have an underestimation of what your nutrients are, we have, in our model, we have an underestimation of the surface nutrients, nitrate in particular, in the Bering, and we see that right away in the sea ice as an impact. So yes, I definitely do see the nutrient limitations there. In terms of grazing, I didn't mention, but we do have 3 categories of phytoplankton three categories of ice algae, diatom, small plankton, and then a Phaeocystis type. And I find that we need very strong grazing on the smaller plankton through heterotrophic grazing. So yes, the diatoms we don't have very much grazing on, but we have strong grazing on the other 2 groups to keep them in check.
All right, great. Thanks very much. So let's move on to Georgina. And she will talk about the ecosystems modeling in the Arctic. Thanks, Nicole. You're still on mute. You're still on mute.
Okay.
There you
Can you hear me now?
Yes.
And you see the slide?
Yes, ma'am.
Okay,
Go ahead.
excellent, great. So a little bit just told you I was gonna talk about ecosystem modeling in the Arctic. That's not the primary. We will touch on that at the end, and I've been using the HiLAT model and looking at the ocean ecosystem dynamics in the Arctic, and I'm gonna touch on that at the end. But the primary focus of this talk is just to bring to you some of the insights that I've learned from past efforts at model data integration from some big integrated ecosystem research projects that I have been involved with over the past decade, which I think there might be some valuable insights that we can incorporate as we're moving forward to do this kind of thing within the ARPC framework. So we're going to talk a little bit about some parameter sensitivity work that we did, talking about what I think are valid ways to use data for model validation. And then I'm going to hand over to my co-author, Clara Deal, who's on this call. And she's going to talk to you a little bit about a white paper that she is getting spun up that'll have a focus on how we can improve model data integration. And Clara's going to tell you about that and solicit your feedback. So what is it? I said IARP, that's an integrated ecosystem research program. And NPRB has sponsored about 3 of these now. They sponsored, the first one was in the Bering Sea. Then we're just wrapping up the Gulf of Alaska one, and they're now sponsoring one in the Arctic too. The unique thing about the Bering Sea and the Gulf of Alaska one is that right from the beginning, they had a very strong modeling component to them. And so they're unique from many other research programs in that the integration between the models and the observational components were envisioned and outlined right from the beginning of the program. So this schematic is basically what the program looked like from a modeler's perspective. So we had this, do you see my pointer? Does that show up on your screen? If I move that around?
Yes.
Yes, okay. So within, this is for the Gulf of Alaska. So we had several different kinds of model for the Gulf of Alaska, going from the physical model through to the biological low trophic level model. Then we had a series of fish models and management models. And then over on the right, we had, these were the different observational field programs. So an upper trophic level component, middle trophic level, and a lower trophic level component. So right from the beginning, as we're starting the program, we envisioned all of the products and linkages that were coming out of these programs and going into the model. So we had all these critical sort of handoffs between data and models and between the different models and the different observational components right from the beginning. And so we sort of went through this whole process. the idea being, because we envisioned these linkages right from the beginning, it was all going to be seamless, and we were going to have much better model data integration than we've had in the past, where you sort of tack on some modeling at the end of a program, for example. So I just thought providing some of the insights that we gain from these programs might be valuable, given that this is something that was envisioned from the beginning here. So maybe we can learn something from the things that went right and the things that didn't go quite as smoothly as we thought. So I'm just going to jump straight to the lessons learned. So then if we get nowhere else, at least we have those nuggets of wisdom for you to take away. So firstly, communication we found to be really key. So these integrated programs had, with the Bering Sea one, for example, had well over 100 different researchers coming from different disciplines, different backgrounds. And for us, having the program managers be heavily involved with this forced communication was critical. And I'd say the programs wouldn't have been successful without that. And it seems silly because so we all know, yes, we should communicate and we need to share our data in a timely fashion. But realistically, without that overarching structure of support, guidance, and logistics, I don't think it would have been nearly as successful as it would have been. So one of the other big, and we did get better at it, as you see. We got much better at talking to each other. One of the big things that came out, and I would say it took a couple years within the program. So the Bering Sea program was 4 years. It took at least that first year for the observational scientists to get over this. There's this great deal of skepticism about what the model is going to, how the model is going to help tackle the hypothesis in the program. And a lot of that was, seemed to stem, we sort of got to the bottom of it, seemed to stem from big confusion about the capabilities of the models and what models could realistically show us. And yes, the model is not going to match your exact pet data point at a certain place and time. But that didn't invalidate the model. So we had, so we've gone through a lot of these discussions. So it's just something to bear in mind as if you're starting a new program with a new set of modelers and observational scientists that haven't been brought together before. It's worth bringing up this kind of discussion really early on in the program, because I think you'll increase engagement of the observational scientists. And then finally, we found that timing presented one of the biggest challenges that we incurred. And because we started the model development at the beginning, when they're getting spun up with the field program, we constantly had a data mismatch problem. And then, as many of you seem to be modelers on the line, it's often, we're not often modeling the current year, we're modeling several years back because of the available forcing data to drive these models. So we consistently had this mismatch in what we could model and what the data was available for. And it was all sort of falling together right at the end of the program. So that I think there's still a lot more that we could do, but now that program is sort of getting wrapped up. But so that's something to bear in mind, and maybe we won't be running into that program, that problem within IARPC, because developed models and existing data sets have been brought together. But that's certainly something to bear in mind. So talking about sources of model uncertainty, and we've touched on this already. There's a lot of observational error that could cause uncertainty in how well we think our models, ecosystem models, are doing. There's also natural variability that the models are not going to be capturing. And then within any model, you have boundary conditions, initial conditions, the actual spatial structure you decide to implement in your model. These all bring in potential errors. But the two that we're going to focus on right now is where I see the biggest areas that we can have linkages with the observational scientists. And that's certainly what we did within the IARPs that I was involved in. So the structural complexity of the model, and the uncertainty in your parameters and how they can help narrow that down. So they're the two components I'm going to touch on right now. So this is the a schematic of the model that we entered the Bering Sea Integrated Research Project with. So that, each box basically just representing a variable within the model, and the arrows are sort of representing the linkages between. So through a lot of these in-depth discussions that we had within the integrated research program, the scientists were actually able to provide us with a lot of feedback as to whether the structural complexity of the model was appropriate and if we could improve upon that. So I would say some of the big successes of that program were that the scientists there had sufficient data and knowledge of the ecosystem that they knew that we could split this microzooplankton component into two separate components, which were biologically different and interacted within the food web in a different way. And then similarly, they did the same with the large copepods. There's an offshore population and an onshore one. So I appreciate that. So this model, because of the nature of the project, and we were coupling to fisheries, it's very sort of bottom end heavy, more zooplankton and the secondary producers. But I would imagine that the same might apply with the nitrates and the phytoplankton for like some of the more BGC kind of models that we've been seeing applied to the Arctic. One sort of downside of this approach, there was a lot of enthusiasm early on in the project to include jellyfish. We have to include jellyfish, and we've been seeing these big blooms of jellyfish in the Bering Sea, and the observational scientists pushed really heavily to include this component. And so we did, because within a model, of course, you can sort of code in anything you like, but to this day, I still feel like that's a very under-validated component of the model. So we had all this enthusiasm early on to include it, and then it turned out, well, yes, we've seen them out there, but actually we don't have the data or know the biological processes that I can code in to actually represent the dynamics of that component really effectively. So that was a lesson learned. So I'm just going to briefly touch on some sensitivity analysis. And actually, Yvette, this is the stuff that you were involved in a long time ago. So this is a sensitivity we did actually on a one-dimensional version of the Bering Sea model that I just showed you. And that's just to understand how, every one of these models has sort of tens to hundreds of parameters behind the black box. And as modelers, we do our best to plug in the values for those. But as you all know, really, most of those parameters have quite a wide range or narrow range associated, which we may or may not know. So with this experiment, Yvette and I actually, ran the 1D model about 3,000 times. And then we were able to rank all of the parameters within the model so on the bottom I'm showing you, and don't worry about reading it, but just to give you the general idea of what we did, I had about 20 different model outputs. One of them was like summer nitrate in the mix layer, or concentration of phytoplankton in July, or sort of things like that that we were interested in. And then on the y-axis, this is all our parameters. And then the darker the color, the more important that parameter was to any one particular diagnostic output. So doing this, it allowed me to focus on which parameters within this model were important and worth worrying about. So as we talk about model data integration, and scientists comes up to you with their favorite data sets, and they have, they can really refine this, one little variable up here that actually isn't showing up as important for any of your variables. We need to think really carefully about, well, are we going to stop and integrate that into the model right now and rerun it, or what is the importance of that parameter? And it also allows us to say, hey, look, we know, for example, that in this model, the phytoplankton growth parameters in particular are really important and affect model output across the board. So if anyone has data that would help me with those parameters, that's where I would spend my time and effort. Okay, so this is a schematic of the Bering Sea. The color blobs that you see, we basically throughout the lifetime of the project, decided that we were going to divide the Bering Sea up into these set geographical regions. And the reason we did that is because it became apparent early on that direct point-to-point comparison of data really isn't that meaningful. So the data that the scientists are going out and observing has a lot of natural variability associated with it. And this particular model is a 10-kilometer model. So it's just unrealistic to expect a model to replicate, for example, the nitrate concentration, at this one particular location at any point in time. And for it not to do so and to judge the model as a failure, we felt like it was too harsh a test of the model. So what we did instead is we grouped the data by months and by regions. And even doing that, we see all this data was collected under the Bering Sea project, but then a lot of it, if you look down this little schematic at the bottom, all of this purple stuff, that was from the Probes project, which was in the 80s. Along the coast, there was a smattering, these little sets here, that was data from the Inner project. So this is all the data ever that we have for the Bering Sea. So at first glance, you might think, wow, this is great coverage. The Bering Sea, we've got a really great data set. But then when we sort of look at, well, how much data do we have for each region by month? So many of the northern regions, we actually only have data that covers up to 3 months a year. Most of the regions do not have any data for January, February, November, December. So that gives you a much better way of looking at what kind of data really is available. Now, because the data is, and those cruises, when they went out on these research cruises, they generally started, I believe, in the southern area, and they kind of progressed their way up. So and that made sense because they were sort of moving up with the ice. But most data points were actually only sampled once, maybe twice. So that gets back to the point that was raised earlier, that most of these data points don't have any standard deviation, no error associated with them. Okay, so because of that, what we decided to do was basically group all of the data by those regions and come up with regional averages. This is I'm showing you for nitrate. So when we did our model data comparisons, we compared the averages for each region. And at this point, we didn't actually even feel like we had enough to break it out by year. So this is really looking at the nitrate climatology for each of the regions. So this is just standard, this Taylor diagram showing you the correlation. So most of the data and observations are correlated reasonably well. And then there's a little bit of a bias in the nitrate. But that's sort of as far as we felt that we could take the comparisons at this point because of the amount of data that was or was not available. And I'm imagining that the situation is going to be even more exaggerated in the Arctic. So that was nitrate. So looking at prime production, we did a similar kind of thing. I'm just going to show you a time series here though. This was I had to group the prime production data for 2 regions on the shelf break. So the blue is my simulation and with its error bars, and then the observed. So you can see that in general, we are capturing that 2-peak trend. There's a little bit of a lag. But again, so we feel like we're kind of getting the climatology okay, but we didn't feel like we had enough data for interannual comparisons at this point. Okay, so what we moved on from then, rather than doing these sort of location comparisons, is to talk about, well, what are the essential features of the Bering Sea that you feel like the model has to capture in order to be recognized as looking like the Bering Sea?
Georgina?
Yep.
Can I interrupt for a second?
Sure.
We're running a little bit short on time, so we only have 13 minutes left or so on this session.
Okay.
Would it be possible to fast forward a little bit?
Fast forward. Yeah, I'll do these next 2, and then I think I can probably transition to Clara. Does that sound good? Okay.
Sounds good. Thanks.
Well, but you always put me at the end and cut me off. It's like the 3rd time. Okay. Okay, so we decided instead of doing these direct comparisons, we wanted to identify features. So the Bering Sea is known for what we call the green belt. So that's a really high production region along the shelf break. So we went ahead and looked to see if the model did that, which we think it did pretty well. Another feature that is really important in the Bering Sea is the subsurface chlorophyll maximum along the 70-meter isobath. So the top one is some observational fluorescence data showing you the subsurface chlorophyll maximum. And then the bottom showing that the model can do that too. Now, at this point, the comparisons are relatively anecdotal. It's like, oh yeah, look, see, it's doing it. So at this point, I'd like to be able to progress that further to something more quantifiable in terms of reproducing these features. So that's the highlight model, but I think we'll skip that today. And then we'll move on to Clara. So basically, I've just given you some ideas about some of the problems we encountered. And we feel like we're making progress because we're moving towards identifying features and perhaps more realistic tests of the model and is it useful. So then I will pass on to Clara now. She's going to tell you how we hope to move forward. All right, Clara.
Yeah, let's see. Hello, everyone. I just have 3 slides here. And to start out with, I'd like you to know I've been charged by IARC, UAF, and Los Alamos to lead the writing of a white paper that will describe the challenges of understanding biogeochemical processes in the Arctic marine system and their climate impacts. So the goal of this white paper is to motivate a workshop that will bring researchers together, observers and modelers. Bring them together so that when it comes to improving process studies in support of model development and improvement, we're not all like reinventing the same wheel. Now, once I have a rough idea what this white paper will look like, I'll ask a group of contributors to provide specific input on the draft. And please let me know if you're interested in being one of these first draft contributors. Next slide. Okay. Well, this white paper will emphasize the need for more efficient model observation integration. So I realize that there are a few researchers who only make observations or only do modeling. Most of us lie somewhere in between. However, one tends to identify with the tools that they're most comfortable with. So in this cartoon, the choice of what we have here are 2 polar animals sitting together on an ice floe. They don't co-occur in nature. And the intent of this is to represent the need for modelers and observers to come together at the same table, regardless of background. And what you see in the second figure here, it's what, from a modeler's perspective, from this paper I was a co-author on, it's what we think a 5-star observer might be. Okay, next slide, last slide. Okay, I'd like to leave you with a preliminary rough outline of what will be in the white paper. The white paper is going to highlight challenges, knowledge gaps, opportunities on the broad topic of Arctic marine and coastal biogeochemical processes in Earth system modeling. That's the broad topic. So what I envision there'll be approximately 3 core group or a core team that will coordinate the development of the white paper. And then we'll ask a group of contributors to provide specific input to the draft. And again, if you're interested in being a contributor on this first draft, please let me know. Or if you'd like to wait and provide input and feedback later on the complete draft, we'll be circulating that or sharing that with the broader community in early 2019. So that's what I had today.
Great. Thanks, Georgina. Thanks, Clara. I'm a terrible timekeeper. We know that all now. So we only have 8 minutes left before the bottom of the hour. So if there are any pressing questions that people want to bring up, then we have a couple of minutes to discuss those.
Yes, this is Guillermo from Bonn. Thank you, all three of you. Great presentations, very informative. One comment and one question for Clara. The comment regarding everybody's presentation is that there was a common mention or need to have better data, more data being available. And that's, coincidentally, that's being discussed today and tomorrow in Germany at the Arctic Science Ministerial. The good news is that one of the co-chairs of the Marine Ecosystems Collaboration Team, along with Danielle and myself, it's Jackie Grabmeyer, who is there representing the US. So that will be a great segue to have her present at the next meeting, joint meeting in November. Data availability and Pan-Arctic integrations are top priorities in many international agendas. And this brings together model and observations, which was just mentioned. And my question to Clara is, are you, in order to develop this workshop, are you bringing or are you connected to the famous group? Because they have go ahead, sorry.
I've participated in famous, well, not in the recent years, but so I have connections to people in these groups, and I've been on a couple of famous papers. So I know that there's a couple, a scientist there who's a modeler or observations in sea ice biogeochemistry, and they're doing a model in your comparison. So I keep up with that. And so I was thinking too, of course, it'd be great to present this at maybe in the next FAMOS or connect with those people, definitely. Yeah.
Yeah, and they have a strong international participation, so that will be great. Yeah.
Right, yeah, I know a lot of those people from being involved. Yeah.
Awesome. Thank you.
Yeah, thank you.
Yeah, I have a comment because for the Arctic, mainly the US is the Western Arctic centric. So if we want data for the other part, we will have to reach out. And when I say Western Arctic centric, even for the model, it's very hard to go over the line with Russia. Mm-hmm. There's also only one program there that they have been collaborating with the Russian, but and I don't know how to solve those problems, but if you look at the Chukchi Sea and the Bering Sea, as well as there are a lot of things happening on that side of the world that influence the ice and the ecosystem. I don't know how to address that problem. It would need a more international collaboration and strong collaboration there that most likely won't be able to come from the scientific community, but more from the program manager from NSF or NASA or those groups. I mean, we can push it, say that there's a need, we recognize it, but I don't know from furthermore how we can do that to gather something.
I think that's been an issue for quite some years.
Right. Clara, Igor and Vladimir may be able to help you. Yeah. IARC researchers that have Russian citizenship. Yeah, I don't know what their connection to biological data is, but
it has been more on the the science researcher to researcher level.
Yeah,
and, it's hard to.
Yeah, well, a few years ago it was open, researcher to researcher, but it's not open anymore from researcher to researcher because researchers who are from Russia are very concerned about distributing data. So I'm not sure how to approach that either the NATO issue or either any other. NATO also was a very big group in participating in the distribution of data at some point when it was just at the Cold War. Then after it was open and now it closed again. And the experimentalists know a lot about that also.
Jackie Grebmeyer.
With our model is that the model doesn't stop at the boundary, but the calibration stops. And then there's the Canadian community that is a lot easier to involve also, they have a lot of data also.
Yeah.
And now the Japanese and the Chinese have a lot of data.
Yeah, and they're working with the DBO program and Jackie Grebmeyer's group. And
Jackie Grebmeyer, yeah. So, I mean, there are a lot of data available. It's just that the two communities have to get together and maybe you're right. I mean, it's hard to keep track where all the databases are. And that's the problem, is that there is not one single database where you can go and grab your data.
Right, and that might be one of the, we can mention that in the white paper as a need, the inventory.
The need is to have a more organized repository that would benefit the community. And I'm sure it's pretty much the same problem if you go to Antarctica.
Yeah, good point.
That's a great comment. So we're at the bottom of the hour, and I guess we have to wrap it up. So